Hello, guys, and welcome back to another lecture. My name is Matthew Belzer, and if you're in my class, as always, I would actually have your lecture handout either available on another digital device so you can type your notes as you follow along with today's lecture, or print it out so you can kind of write out your notes and answer the questions that you've been provided on that lecture handout today. So today we're going over histology. Histology is the study of tissues. And at this point in the class, a lot of students ask, well, why are we doing this? Why aren't we talking about organs yet and organ systems? I feel like I would be better at that. And you can't really understand organs and organ systems until you understand what physically makes them up. So if you think about how all anatomy and physiology classes are going to be taught, they're going to be taught along levels of organization. So when you think about the way we've gone over this class, we had our intro material, and that intro material was familiarizing you with the language of anatomy and the body structure and the compartments and the different cavities and membranes. And then we shifted to talking about general chemistry, the very small stuff. From general chemistry, then we kind of expanded and we talked about biochemistry. And what we're doing here is we're going up our levels of organization. Right? So we started with the least complex level of organization, and then we shifted our focus to cells. Cells are the building blocks of living things, of multicellular organisms. And we talked in great detail about cells. And then you start putting together trends. You know, what happens at the cellular level can definitely impact what happens at the system level, meaning the entire organism. Now, groups of cells working together to carry out a common set of functions makes up a tissue. So all multicellular organisms are composed of different tissues knit together in certain, three-dimensional arrangements. For example, or humans in general, for example, are just a constellation of epithelial tissue, connective tissue, muscle tissue, nervous tissue. And all that tissue kind of gets knit together and interacts with other tissues in this extraordinarily complex three-dimensional arrangement, and that's what makes a human. So I'm physically composed of tissues, you're physically composed of tissues, and in order to understand anatomy and physiology in any degree of detail, you have to understand that, hey, we are comprised of tissues. So understanding tissues gives us tremendous insight into what's happening at the higher levels of organization. So the key points of today's lecture are going to be mapping out different tissues, and then we're going to draw our focus to epithelial tissue recognition and naming conventions. So we're at follow along 1 now. You can kind of track along with your handout, and I'm going to make some comments about the handout for the people in my class, so as you're following along, you know how to not get lost. So follow along 1, histology. Histology is the study of tissues. Why do we care about histology? Well, histology is really important to the study of pathology. Pathology is the study of disease. A pathologist is a disease expert. So pathology is the study of disease, and pathologists, for example, when they take your blood, utilize knowledge of general chemistry and biochemistry. When they're doing a pap smear and they're looking at the shape of cells and the staining patterns of cells to make educated guesses about the presence of cancer, they're utilizing cytology. When you take a biopsy, let's say that you have a suspicious lump in your breast, and they take a chunk of that. So they take a needle and they take a little biopsy of it. In order to understand what's happening and to be able to evaluate that and make diagnoses, you need to have an understanding of histology. And essentially, if you understand what tissues should look like, you'll be able to identify when tissues have shifted or changed their appearance, which could be reflective of a disease of some kind. So pathologists use tissue histology all the time, and because you guys are getting into the professional world and you're going to be allied health professionals, it's good to understand a little bit about why we study this. So let's look at how a pathologist could use histology. Here we have a biopsy. So let's say you have a pediatric patient come in. They're 9 years old and they have this painless muscle weakness. So their muscles are just getting weaker and weaker, and they're tripping over things and they're having problems. So their parents bring them in and go, I don't know what's going on. I mean, their muscles look like it's the same size. I haven't seen their muscles atrophy or shrink, so I can't figure it out. You could do a few things. You could do a genetic test. you could do a genetic test and follow that up with a biopsy of a muscle. So here we have healthy skeletal muscle tissue in a cross section. This is what skeletal muscle tissue should look like. Now, you don't have to be a pathologist to recognize that this is a little bit different, and this change in the tissue is reflective of a disease called Duchenne's muscular dystrophy. And what happens in Duchenne's muscular dystrophy is there's a gene that's mutated that codes for a protein called the dystrophin protein. And dystrophin essentially stabilizes skeletal muscle tissue when it's working. So when you exercise, for example, that dystrophin protein locks that skeletal muscle tissue into place and prevents it from being broken down. If that protein isn't working properly, whenever you use that muscle, any damage to that muscle is ultimately going to result in that muscle dying, those muscle cells dying off. And what ends up replacing that is fat or adipose tissue. So adipose tissue is very different than muscle tissue, and when you see this fat kind of perfuse through the muscle, that is a telltale histological feature or pathohistological feature of Duchenne's muscular dystrophy. So if you know what something looks like when it's what it's supposed to look like normally, which is what we're doing with the study of histology now, then later on down the road you can start to identify deviations from the norm that could be reflective of disease. Now, when you think about the tree of medicine, I always like to show this just so you kind of understand where pathology exists within the entire study of medicine. So down here we have things like anatomy, embryology, histology, chemistry, pharmacology, immunology, physiology, microbiology. These are the things that you study at the 4-year or 2-year universities as you're building up to become, let's say, a health professional or a professional in the field of science or a medical professional of some kind, right? So right now we're studying histology, and all of these different studies kind of form the roots of medicine. These give you the base knowledge that you need. All of these feed into pathology. Every single one of them feed into pathology, which is the study of disease. And then pathology branches out into all the different fields of medicine. So it doesn't matter whether you're working as a dermatologist, a psychiatrist, a neurologist, a surgeon, as a pediatric medical professional. Pathology forms the root of what you do. For example, if a kid comes in and they're, feeling faint and you can't figure out why, you take a blood sample and you send it to a pathology lab to maybe run a chem panel to see if their electrolyte concentrations are off. All of that ultimately filters down into general pathology, and understanding histology gives us a bit more of a foundation in how we understand medicine as a whole. So the pathological paradigm, the gross anatomy, microscopic anatomy, and biochemical structures of cells and tissues provide information about disease etiology. Etiology is a fancy way of saying what's causing a disease or what's causing a condition. So it allows you to make diagnosis. It allows you to determine the degree of deviation, the grade. Is it mild? Is it severe? It allows you to determine the extent or the stage, right? Is it isolated to one region of the body? Is it all over the place? So we've all heard of staging, for example, with cancer. And I'll give you just a quick rundown of what I'm talking about. Like stage 1 cancer is a small tumor in one region of the body, and this gets much more complex, right? Stage 2 cancer is a larger tumor in one location of the body. Stage 3 cancer is when that cancer is spread to the lymph nodes. Stage 4 cancer is when it's spread or metastasized past the lymph nodes. And all of those diagnoses are done using histology as a basis. So let's give you a practical example. Morphological diagnosis just means diagnosis based on anatomy. So you have a patient come in and they're complaining of persistent heartburn, right? And you go, okay, I listened to their chief complaint and I think they have a disease called gastroesophageal reflux disease. So what I want to do is I want to do an endoscopy and I want to take out a little sample of the lining of their esophagus to look for changes. So as a pathologist, you know that the esophageal lining should look like this, right? Now, you don't have to be a pathologist. Let's say you do this on your patient and you notice that the lining of their esophagus looks like this. You see all these little inflammatory cells, and you know what that means, and you go, oh man, yeah, those changes are consistent with gastroesophageal reflux disease. So now the symptoms and the signs, the things that you can physically measure or see through the biopsy, both support the diagnosis, so you can be more confident in the diagnosis you're making. If you ever have a patient who's getting a biopsy, like let's say you work in oncology as an oncology nurse and a patient gets a biopsy and they're like, why is it taking so long? Yeah, that can be frustrating. It's probably because the pathologist or his assistant or her assistant is sitting down in a lab with 5,000 biopsies to look at, and they have to look at them very, very carefully. So there can be a little bit of a backup, and that can result in delays in getting like test results. So when you think about orientating ourselves to the tissues of the body, a group of cells working together to carry out a common set of functions makes up a tissue. A group of tissues working together to carry out common sets of functions make up organs. Groups of organs working together to carry out common sets of functions make up organ systems. And the first organ system that we're going to look at after we go over histology is going to be the integumentary system. For now, we're going to draw our focal point to tissues. Now, don't start filling in those boxes yet. I've given you 4 and 4. Today's lecture is going to be dedicated to epithelial tissue, so I'll tell you when to start filling in those boxes. Don't start filling them in yet. So we have epithelial tissue, connective tissue, muscle tissue, and neural, aka nervous tissue. These are your 4 primary or basic tissue types. Each one of these can be subdivided into specific types of tissue, right? And that's what we're going to focus on today with epithelial tissue. So our focus is really going to be on epithelial tissue. So when you think about epithelial tissue, and this is follow along too, so you can kind of track with me, epithelial tissue is a covering and lining tissue. It covers and lines any surface that's exposed to a free space or a lumen, and I'll explain what that means. So when you look at me right now, my skin, the outer layer of my skin called my epidermis, is made up of epithelial tissue, and that epithelial tissue essentially forms a barrier between the external and internal environment of my body. It protects underlining tissues. So when I say covering and lining, epithelial tissue covers and lines my body, covers and lines things like my oral cavity, the rectum, the anus, right? In fact, all your hollow organs, things like your GI tract, your reproductive tract, your urinary tract, all of those tubes are lined by epithelial tissue. So we're going to talk about what a lumen in a tube is right now. So when I say the lumen is lined by epithelial tissue, let's pretend this tube is a blood vessel or a duct, not a duck, D-U-C-K, a duct. D-U-C-T, a tube in the kidneys, or whatever it is, right? Or the GI tract, it doesn't matter. So if this was a blood vessel, we'll just use blood vessel, and I said, where would the blood flow through the blood vessel? You'd say inside the tube. Well, inside the tube, this area is called the lumen of the tube. And this lumen, the internal portions of this tube, would be lined with epithelial tissue. All of the tubes in your body, and you have miles of tubing in your body, all of those tubes, the lumens of those tubes, are lined by epithelial tissue. So it covers and lines everything. And it can provide a constellation of functions. It can be a smooth surface, so for example, blood can flow. It can be a protective barrier like your skin. It can function as filtration barriers or diffusion barriers like the sacs in your lungs. So it's a covering and lining tissue. The other thing epithelial tissue does is it forms glands. So whenever you say a gland, like a sweat gland or one of your endocrine glands, like the adrenal glands, and you're wondering about what actually secretes or produces the secretion, whether it be sweat or a hormone, epithelial tissue forms the secretory portion of glands, meaning that epithelial tissue is what actually is producing the secretions of whatever gland it is you're talking about. So that's a general overview of the functions of epithelial tissue. Now, a question I always ask on an exam, and if you're tracking along using your handout, I would definitely put an asterisk by this, especially as a short answer question, is I always say, describe the 6 general characteristics shared by all epithelial tissue. So what are the 6 general characteristics shared by all epithelial tissue? Because there are 8 epithelial tissue specific types that you're going to need to know. But what do all those epithelial tissue types that you need to know, what do they all have in common? So let's look at this. So here we're looking at a duct in the kidney, right? This tube in the kidney. And here we have the lumen. That's where the fluid is going to be flowing. And lining that duct, we have epithelial tissue, right? So these cells working together to carry out a common set of functions that are going to line the entire length of the lumen of this tube. So it's 3-dimensional. It's weird teaching anatomy because anatomy is very 3-dimensional and we teach it in 2 dimensions. We have this tube and it's lined by cells, epithelial cells, making up epithelial tissue. Now, when you look at these cells have a top and a bottom. So the top of a cell that contacts or comes into contact with the lumen, or what I sometimes call the free space, or the fluid cell interface, or the external environment if you're talking about your skin cells, right? That top is called the apical side. So the top of the cell is called the apical side, and the top of the cell serves a very different function than the bottom of the cell. So whereas the top of the cell interacts with this space, it could be the lumen or the external environment, the bottom of the cell is there, it's called the basal side of the cell, it's there essentially to latch that cell down to the underlying connective tissue. Epithelial tissue always latches to connective tissue, that's what it's always anchored by. So if you're thinking like the tiles on your floor, the tile would be like an epithelial cell, the concrete in your floor would be like the connective tissue. Epithelial cells always latch down to connective tissue. The fact that the cells have different sides that serve different functions, you refer to that as being polarity. So if I ask you and you just put polarity, cellularity, and you don't give any description, right, that's a 1 out of 10 response. I want you to explain to me what polarity means. Well, it means that the cells have an apical and basal side, and those different regions of the cell serve different functions. The apical side interacts with whatever the fluid or the surface is, right? Out here, it could be an absorptive surface or it could be secreting stuff, as opposed to the basal side, which is intended to lock the cell down. Epithelial tissue displays what's called cellularity. These cells, if you notice, are right next to one another. That's not the same in every type of tissue that we're going to go over. So close are these cells that they actually form attachments called junctions. In other words, the entirety of epithelial tissue, or the overwhelming majority of epithelial tissue, is made up of cells. It's made up of epithelial cells. That's not the same with all tissues. So if you see tissues are really close to one another, that property, like actually physically touching one another, that property is called cellularity. And epithelial cells are so tightly associated with one another that they form these connections to one another that we call junctions, that we're going to talk about in just a bit. Epithelial tissue is avascular, meaning without blood vessels. So you don't see blood vessels coming up into the epithelial tissue. All the blood vessels that provide, for example, oxygen and nutrients that allow epithelial tissue to get rid of waste, they all run through the underlying connective tissue, right? The epithelial tissue itself doesn't have blood vessels. So it's said to be avascular, so it relies on that underlying connective tissue for all of its nutritional needs and all of its gas exchange needs. So when you cut your skin, like if you get a paper cut, my brothers used to give me paper cuts for fun because they're jerks. But if you get a paper cut, right, and it doesn't bleed, that means that cut is relegated to the epithelial tissue. If it bleeds, it means, if you have a cut that bleeds, it means you've broken through the epithelial tissue and you've gotten to the underlying connective tissue. Now, all epithelial tissue is locked down to the underlying connective tissue by a layer of proteoglycan glue or protein glue called the basement membrane. So we're going to see that a lot today. And Even though you can't see this, one of the functional characteristics of epithelial tissue is that it's regenerative. That means those cells are rapidly dividing, they're undergoing mitosis like crazy, or they're mitotically active, and they're constantly replacing themselves. So when you think about your epidermis replaces itself every 28 to 40 days. Lining of your GI tract, all the epithelial cells lining your GI tract, like every 7 days. These cells are dividing like crazy. They're constantly replacing themselves. I just sloughed off like 1,000,000 cells. Most of the dust in your house is just dead skin cells. So you're constantly replacing those cells. Another characteristic I want you to be familiar with is called innervation. So here you have the epidermis, and the epidermis is made up of a type of epithelial tissue called keratinized stratified squamous epithelium. And the epidermis is what's said to be, and a lot of epithelial tissues are what are said to be innervated. That means that they actually have sensory receptors in them. And in this case, we have free nerve endings, which are responsible for detecting pain and temperature. So if you have like a needle that pokes you, or you put an ice cube on your skin, these sensory receptors detect that change, let's say in temperature, and they send an electrical signal along this neuron, which becomes part of a nerve, all the way to your central nervous system. So epithelial tissue is innervated, meaning that it has sensory receptors located within it that are linked to the nervous system. And that's how information, for example, goes from your integument to your brain. It starts at that sensory receptor. So those are the 6 general characteristics shared by all epithelial tissues. I always ask that as a short answer question because I just want you to orient me to what you're talking about. Do you know why we're calling these things epithelial tissues? What are those characteristics that all epithelial tissue has in common? Now, again, we're teaching something 2-dimensionally that's 3-dimensional. And on your sheet, you're kind of following along and you're looking at the cells that are latched to one another. You have a nice image. So you're not going to have to understand the anatomy in a tremendous amount of detail, but I want you to look at the position. So here's the apical side of the cell, here's the basal side of the cell locked down to the underlining basement membrane. Now, tight junctions. Tight junctions are these proteins that are kind of like shoelaces that weave through one cell and its neighbor or its adjacent cell, and it attaches them, very, very tightly. And what it does is it seals the neighboring cells together so material can't get through the little gaps between the cells. So those are called tight junctions. When you think about tight junctions, have you ever heard the term the blood-brain barrier? The blood-brain barrier isn't like a layer of fat that just extends across the cervical region and prevents, material from getting into your brain. It's actually the tight junctions in the epithelial lining of the blood vessels in the brain, right? And it prevents material from getting into the blood from getting into the brain. Right? So it prevents the passage of material. In fact, if you hear blood anything barrier, blood cerebrospinal fluid barrier, blood testes barrier, you can assume that that's epithelial tissue held together via tight junctions. So those junctions are really important. We have adherens junctions, which are kind of like zippers, and they are strong attachment points between one cell and another. Notice that tight junctions are closest to the apical surface. Adherens junctions typically come next, and they're kind of like protein zippers. They're really, really strong junction that join the actin or the cytoskeleton of one cell with its neighbor. Desmosome junctions are the strongest of the junctions we're going to go over, and they also link one cell to its neighbor, and specifically the cytoskeleton inside. So if you've ever had a sunburn and you've peeled your skin off in sheets, all the epithelial cells in your skin are attached to one another via desmosome junctions, and that's why it comes off as a sheet. Then you have gap junctions. Gap junctions are also called communication junctions. They're holes, they're little channels that extend from one cell to the next, and they allow the passage of materials like ions or chemical signaling molecules. What they really allow is one cell to communicate with its neighbor. right? So when you hear gap junctions, those are allowing cells to communicate. And you don't think of communication as passing, sodium ions from one cell to another, but at the cellular level, that's how cells communicate with one another. They communicate using chemical signaling. And then we have hemidesmosomes. So the image that I've given you doesn't have a hemidesmosome, and I ask you to draw that in. Hemidesmosome means half button. And what hemidesmosomes do is they attach the epithelial cell to the underlying connective tissue or basement membrane. And that's what they do. They're kind of like, if you were to glue two things together, they don't glue one cell to another. They attach the cell to the underlying connective tissue. That's what hemidesmosomes do. So be familiar with those junctions. Now we're going to start talking about Reckitt, which is key point 2 in this lecture, which is recognition of specific types of tissue. So now we're at key point 2, follow along 3. Now don't fill out those little blocks in your handout quite yet. I'm going to explain to you how to fill those out. Before I explain to you how to fill those out, we're going to go over a couple of different question sets. So whenever you're looking at a tissue, first, how do you identify it as being epithelial tissue or epithelium? Epithelial tissue is the adjective form of the word. It means pertaining to epithelium. Epithelium is the noun form of the word, and that means the thing itself. So some instructors are kind of picky about whether you write epithelial tissue or epithelium. I'm going to stick largely with epithelium, and that'll make sense in just a moment. So how do you determine that this is epithelial tissue or epithelium? Well, let's think about our general characteristics. 1, are the cells close together? Yes, they are. That's called cellularity. 2, is there a free space? Meaning, do the cells display polarity? So does one side of the cell, come into contact with some lumen or free space, something that's not cellular in nature, like a fluid interface or an air interface, right? So is there a free space? Yes. So the cells are close together and there's a free space. That's enough to let you know that that's epithelial tissue right there. Another telltale indicator is the presence of that basement membrane. You see all 3 of those things, you go, oh, that's epithelium or epithelial tissue. So those are general characteristics. Now we're going to start naming specific tissues, and we're going to go through a sequence of rules for naming these tissues. So we're going to go over the rules for naming these tissues, and then I'm going to give you questions that are going to crop up on the exam that you're going to take. So the first thing you ask yourself when you are identifying a specific type of epithelial tissue is you say, how many cell layers thick is it? If it is 1 cell layer thick, it is simple. If it is multiple cell layers thick from the basement membrane to whatever the free space is, it is stratified. So you go, okay, simple versus stratified. Then you look at the shape of the cell. So if it's elongated and it's wider than it is thick, we refer to that elongated kind of fried egg shape as being squamous. If it's shaped like a cube, we call it cuboidal. And if it's shaped like a column, we call it columnar. So when I'm asking you to identify specific tissues indicated by the pointer, let's identify this specific tissue first. So I say identify the specific tissue indicated by the pointer. How many cell layers thick? From the basement membrane, it is 1 cell layer thick, so it is simple. Then I ask, what are the shape of the cells? Well, the cells are elongated or shaped kind of like fried eggs, meaning wider than they are thick or kind of flattened. Therefore, they are squamous. And because we see all the characteristics of epithelial tissue, like cellularity, basement membrane, apical and basal sides of the cell, right? We know that it's epithelial tissue or epithelium. So when you're naming a specific tissue, you go, this would be simple because it's 1 cell layer thick, squamous because you have those flattened cells, and then you always tag on the primary or basic tissue type at the end, which is epithelium. Simple, squamous epithelium. What are two anatomical justifications for why you called this simple squamous epithelium? I called it simple because it's a single cell layer thick. I called it squamous because the cells are flattened or shaped kind of like fried eggs. And I called it epithelium because it has all of the characteristics, general characteristics of epithelial tissue. Focus on the first two for anatomical justifications. The number of cell layers when I'm asking about specific tissues, the number of cell layers and the shape of the cells. So identify the specific tissue indicated by the pointer. This would be simple cuboidal epithelium. Identify the specific tissue indicated by the pointer. This would be simple columnar epithelium. So those rules make sense. Now, a variation on simple is actually pseudo-stratified. Pseudo-stratified means falsely layered. And pseudo-stratified epithelial tissue has really, really long cells, and the nuclei are staggered, so it looks like it's multiple cell layers thick, but it's really only one cell layer thick. Now, so questions you ask yourself: is there a free space? Are the cells close together? It's epithelial tissue or epithelium. Then you say, how many cell layers thick and what shape are the cells? That allows you to identify the specific tissues you're looking at. And what we're going to do today is we're just going to go through each of the tissues you need to know, and we're going to ask a set of questions about them. So now you start filling in your boxes. So you have epithelial tissue, and epithelial tissue is broken into two primary categories: simple and stratified. Is it a single cell layer thick or is it multiple cell layers thick? So the simple epithelium that you need to know, first box, simple squamous epithelium. Make sure to read where you find them. Found in lining of blood vessels, which is called endothelium, and anywhere where filtration or diffusion needs to occur rapidly in the lungs and the kidney. Simple cuboidal epithelium makes up ducts and glands. Simple columnar epithelium lines the GI tract. Pseudostratified ciliated columnar epithelium lines the respiratory tract. What we are going to do for the next few slides is look at each one of these individually and think about all the questions that could be asked. A great way to study for this particular unit of the exam, because if you were in class, I would ask you, you'd have to know these, be able to recognize them on the screen, be able to spell the words, and you're going to have to know that taking this online, because I've put in preventive measures to protect from cheating. So this is a really rote memory intensive task. If you were using index cards, because I think flashcards are a great way to study, Quizlet is a great study tool. Ask yourself these questions. Can you identify the primary tissue type, the specific tissue type? Can you give me at least two anatomical justifications for why you called this specific tissue a specific tissue? Where in the body would you find it, and what function does this tissue serve? So we're going to look at each of these tissues just one by one. Practical time. If I said identify the specific tissue indicated by the pointer marked 4, I go, okay, cells are super close together, definitely, and there's a free space, definitely epithelial tissue. So now it's asking not for the primary tissue type, but for the specific, or for the tissue type. I'll say primary if I just want epithelial tissue. You're not going to get very many of those questions. So I look at this and I go, okay, single cell layer thick, and the cells are kind of flattened. That is simple squamous epithelium, right? That's the specific tissue. Follow-up question, provide two anatomical justifications. Well, the tissue is one cell layer thick and the cells are flattened or shaped like fried eggs. Those are two great anatomical justifications, right? For why you called that simple squamous epithelium. Then, where would you find it? You find simple squamous epithelium, one, wherever you need really rapid diffusion to occur. So what you're looking at here, the sacs of the lungs called the alveoli. And what the alveoli do is they allow gases to move across their surface really quickly. So when you inhale, you're filling up these little sacs with air, and specifically oxygen-rich air. So air is moving into these little sacs, and you need to exchange that with your blood vessels, which, right, you can see a couple of them on this particular image. You need to exchange that really quick. And remember, this is three-dimensional. The alveoli are spheres, so we're looking at a cross-section. So this nice thin tissue is really well adapted to that because it allows gases to diffuse quickly because it's not a really thick tissue. It reduces the diffusion distance. Other areas where you find simple squamous epithelium are the filtration membranes of the kidneys, lining blood vessels. When you find simple squamous epithelium lining blood vessels, it serves a slightly different functional role than allowing for diffusion to occur. So think about that. Identify the specific tissue or the tissue indicated by the pointer, right? Provide anatomical justifications. Where would you find it? Now, lining blood vessels, what these pointers are pointing at is simple squamous epithelium. And simple squamous epithelium, when it lines blood vessels, is called endothelium. And that endothelium just provides a nice, smooth, friction-free surface. So simple squamous epithelium can also be nice and smooth in order to prevent things like platelets from catching on the side of blood vessels and triggering a clot. So you're looking at a vein here under a microscope, albeit a very small vein, and you see each one of these? These are blood cells. These are red blood cells you're looking at here. Every one of those is a red blood cell carrying oxygen around. So we're looking at a bunch of red blood cells in a blood vessel. You want blood flowing through a nice smooth surface. So where would you find simple squamous epithelium? You'd find it in the sacs of the lungs. What does it do there? It allows diffusion to take place quickly. You find it in the filtration membranes of the kidneys. What does it do there? It allows diffusion to take place quickly. You find it in the lining of blood vessels. What does it do there? It provides a nice, smooth, friction-free surface. So now let's go ahead and move on to our next type of simple epithelial tissue that we need to know, simple cuboidal epithelium. So up here we have the simple cuboidal epithelium. We can kind of see the three-dimensional arrangement. We see the basement membrane here, and then we see the underlining connective tissue. So when you're looking at this, I will either just have a pointer on it, or I will put a box around it to kind of give you a little hint as to shape. And I'll say, identify the specific tissue indicated by the pointer. Single cell layer thick. This is a duct, by the way, a tube in the kidney. So single cell layer thick. We see our basement membrane, and we see that the cells are really close together. We know it's epithelial tissue. Free space, cells are close together. There's your basement membrane right there. So if I asked for the specific structure, not the tissue, and I'm pointing at that, that would be the basement membrane. So the specific tissue is simple because it's one cell layer thick from the basement membrane to the free space. Cuboidal because each cell is relatively cube-shaped. And now you may go, well, that's more of a circle. There is no simple circular, right? Nature doesn't care what we call it. We've created these conventions to make it easier for us to understand. So it's never going to correspond exactly with what you would expect. It's roughly cube-shaped, right? And then epithelium. So simple cuboidal epithelium, single cell layer thick, cells are shaped like cubes. Where would you find it? You find simple cuboidal epithelium lining the ducts in the kidneys. And what it does there is it allows for reabsorption and secretion. That's essentially what the little ducts in the kidneys are doing. They're reabsorbing material you need and secreting material you don't. Reabsorption and secretion. That secretion function becomes really important because simple cuboidal epithelium makes up the secretory portion of all glands. So any gland you name, sweat gland, thyroid gland, adrenal gland, the glandular pancreas, all of the secretory portions of those glands, meaning the stuff that's actually producing the hormone or the sweat or the saliva, all of the secretory portions of glands are made of simple cuboidal epithelium. So this isn't a gland right here, but it's really good at moving fluid from the cell to the free space. And it could be a duct, and then that duct could carry like sweat to the surface of the skin. So simple cuboidal epithelium, really, really important. Identify the specific tissue indicated by 1. Where would you find this tissue in the body? What functional roles does this tissue serve? Identify the specific tissue indicated by the pointer marked with my hand. Think about that. Google Image is your friend here, by the way. And this is kind of nice because you can see the blood vessels. So when I say secretion and absorption, right, you're absorbing material from the ducts in the kidneys, because this is a kidney we're looking at. In fact, the kidneys are just big bundles of simple cuboidal epithelium for that matter, and connective tissue. We reabsorb stuff we need, like water, electrolytes, and nutrients, and we reabsorb that stuff back into the blood. And then we secrete stuff we don't need, for example, toxins, and we secrete that into these tubes. The fluid that's left in these tubes at the very end of this pathway ultimately becomes urine that we get rid of. Also, you find simple cuboidal epithelium in all glands. Simple columnar epithelium. So let's tie the tissue to the entire organ here. You have the GI tract, and the GI tract is broken into different segments like the esophagus, the stomach, the small intestines, the large intestines. If we were to take a chunk of the GI tract from the esophagus all the way to the rectum, what lines the cells of the GI tract, right? So you have the GI tract, which is well adapted for absorption. Here's your small intestines. You have these finger-like projections that are histological features of the small intestines, or folds, right, called villi. And lining these villi, there are these little cells, and the cells that you see are simple columnar epithelial cells, and oftentimes they have microvilli on their apical side. Microvilli are different than cilia in the fact that microvilli are anatomical adaptations to increase surface area for absorption to occur. So microvilli are anatomical adaptations for absorption. So lining the cells of the GI tract, you find simple columnar epithelium. So if I was to ask you, identify the specific tissue indicated by the pointer, it's simple because it's one cell layer thick. It's columnar because all the cells are shaped like columns. And notice the nuclei don't line up perfectly, but they line up pretty well. And it's epithelium for the reasons we've discussed. Where do you find this? You find it lining the GI tract. What's it well adapted for? Well, it protects this underlying connective tissue pretty effectively, but because you see that little hazy layer right there, that's microvilli. What you're looking at are microvilli. They're very, very small. And they're adaptations for absorption. What simple columnar epithelium is very good at doing, when you eat something, you break it down to its smallest components, like starch into glucose, proteins into amino acids, and you have to absorb that material from the lumen of the GI tract, which is where the food stuff would be, right, across this epithelial tissue into your cardiovascular system, into your blood, and then it's distributed throughout your body, and we use that stuff. So here are the villi, and you can see the underlying connective tissue. Connective tissue tends to stain lighter than epithelial tissue because it's not composed entirely of cells, and cells stain darker. So what you're looking at here is simple columnar epithelium. It's really obvious when you see the lining up of the nuclei. When I show you the staggered nuclei, you're gonna go, oh, okay. So it's simple because it's a single cell layer thick. Columnar because the cells are shaped like columns. Where do you find it? Lining the GI tract. What is it well adapted for? Absorption, right? The absorption of material. And that little hazy stuff you see on top is not cilia, it is microvilli. Microvilli are anatomical adaptations for absorption. You don't have to put that in your answer, but if I was to ask you a question on top and say, hey, what are those cells lined by? It'd be microvilli. They're smaller and they serve a different role than cilia. So microvilli are anatomical adaptations for absorption. Cilia, on the other hand, are anatomical adaptations for moving material around. So this tissue, which is one of my favorite tissues, is called pseudostratified ciliated columnar epithelium. You have to include all 4 of those words in order to get 100% or even to get the answer correct at all. And this is memory intensive. Now we're into the rote memorization, and what we're really evaluating is, can you remember these things? Because later on, when you take your credentialing exams, like your NCLEX, for example, you have to remember your 200 medications, what they do, what the side effects are, when you use them, etc. Learning large amounts of material and having that information readily available to you and not needing to Google it is one of those things that we want our clinicians to have. So when you look at this, it's a single cell layer thick, and here we have the basement membrane. Now it doesn't look like a single cell layer thick because these nuclei are staggered all over the place. It is obvious when the nuclei are staggered. It is obvious when the nuclei are staggered. So that's what the pseudo-stratified is. It's really one cell layer thick, but it looks like multiple cell layers thick. It's ciliated. We have these hairs and the apical surface of the cell, and these cilia are responsible for moving materials across the top of them, notably mucus, right? Columnar, because each of the cells is shaped like a column. They're shaped kind of like those air dancer things. And then epithelium for all the reasons that it's epithelial tissue. Where do you find pseudostratified ciliated columnar epithelium? You find it lining the respiratory tract. So like the trachea, the bronchi. What does it do? Well, you have these ciliated cells that rhythmically beat and they move mucus across their surface. It's called mucociliary clearance or the mucus elevator. So when you inhale things like dirt, debris, pathogens like COVID, they get caught in this mucus, and then the cilia physically move that mucus up and out of the respiratory tract. Now, embedded within this pseudostratified ciliated columnar epithelium is a type of cell that becomes very important. And these cells that are shaped kind of like these chalices or goblets that don't pick up stain very well, so they have kind of a translucent appearance, these are called goblet cells. Wherever you see these kind of translucent chalice-shaped cells or cup-shaped cells, they're always going to be producing mucus. Mucus just doesn't pick up stain very well, but these are just little mucus-producing factories. So where does the mucus for that mucociliary clearance come from, or that mucus elevator, all that mucus lining your respiratory tract? Comes from these goblet cells. And that's real important, man. You don't think about, all of the things that your respiratory tract is exposed to, and most of them get caught in this mucus elevator and get moved up, and you either swallow them or you spit it out when you go like, right? So it's really important. These goblet cells are really important. Now, when you're thinking about mucociliary clearance, here is a live image of the pseudostratified ciliated columnar epithelium lining the respiratory tract, and all of this is mucus, and that's all the stuff that's caught in that mucus. So you don't think about the fact that you inhale 10 fungal spores every time you inhale. You inhale a constellation of bacteria, dirt, debris, cells, particles. But it becomes really obvious how much you're actually inhaling and exhaling when you look at what gets caught up in that mucus. So that cilia is physically moving that mucus across it. Thank you, Harvard, for that image. So that's what cilia does. It's not an adaptation to increase surface area for absorption. It's an adaptation to move fluid across its surface. And when you look at cilia, this is an individual cilia. It's connected to these proteins that are formed from the cytoskeleton. When I say proteins are like little molecular machines, I mean they're like little molecular machines. They do stuff. So you have a gene for the proteins that make up this component that allow the cilia to beat and physically move materials across it. Can you have genetic disorders that affect these proteins that move cilia? Sure you can. Now, stratified epithelial tissue you need to know. So wherever you're at, you're probably trying to figure out specific tissues. I did not put them in order by any stretch of the imagination. Now go up to that handout, and these are the 4 types of stratified tissues you need to know. We have non-keratinized stratified squamous epithelium, keratinized stratified squamous epithelium, stratified cuboidal epithelium, and transitional epithelium. So, non-keratinized stratified squamous epithelium. Stratified because the tissue is multiple cell layers thick. Squamous because the most superficial cells, not the deep cells, but the cells closest to the surface, are flattened and elongated. So stratified, multiple cell layers thick. Squamous, most superficial cells are flattened or elongated. And epithelium, for all of the reasons that it's epithelial tissue. The cells are tightly packed, there's a free space, basement membrane, etc. Now, how do you distinguish between keratinized and non-keratinized? You come up here to the most superficial cell layers. If the most superficial cells still have nuclei, that means those are living cells, metabolically active living cells, and that would be non-keratinized. So where do you find non-keratinized stratified squamous epithelium? Wherever you have a lot of mechanical or chemical stress that isn't subject or prone to water loss. So wherever you have a lot of mechanical stress that isn't prone to water loss, lining your oral cavity, lining your esophagus, lining the vaginal canal, lining the rectum and the anus. These areas are subject to a lot of mechanical stress, right? Like when you chew a Doritos chip, you don't want to rip up the side of your mouth. When you drink Mountain Dew or poison, you don't want to burn away the tissue and get to the underlying connective tissue, right? Your mouth would start bleeding. So they're found in areas that need a lot of protection that aren't prone to water loss. So think about all of those areas that I just talked about. It's just tough stuff. It doesn't tear easily. Protects that underlying tissue just because of the layering effect. So that's non-keratinized stratified squamous epithelium. Think about this tissue, identify the specific tissue indicated by the pointer, provide me 3 anatomical justifications. So anatomical justification 1, it's multiple cell layers thick, that's why it's stratified. Anatomical justification 2, the most superficial cells are flattened or elongated, that's why it's squamous. Justification number 3, the most superficial cells have nuclei, meaning that it's non-keratinized. Where do you find this? Areas, right, that are subject to a lot of mechanical or chemical stress, but aren't prone or susceptible to water loss. Like your oral cavity, not a lot of water gets lost there, right? It's internal. It's not exposed to the external environment as much, and therefore evaporation doesn't occur quite as effectively. So when you think about this, the lining of the esophagus, using that example of from the very beginning of the class when we were talking about how you use histology, people with gastroesophageal reflux disease, sometimes you can actually see the acid wearing away the different layers of the cells, and this would be non-keratinized stratified squamous epithelium lining this here, so you can actually see that epithelial tissue or lining wear down as it's exposed to acid, and if it wears all that non-keratinized stratified squamous epithelial tissue down, it'll start bleeding, because you'll get to the underlying connective tissue. Now, keratinized stratified squamous epithelium. Keratinized stratified squamous epithelium makes up the epidermis. So the outer layer of your skin is called the epidermis. Everything you see when you look at somebody is essentially keratinized stratified squamous epithelium. It makes up the epidermis all over the place. So how do you identify it? Well, here's the underlying connective tissue of the dermis, and then you look at this. Let's say a bracket's around this, and I say identify the specific tissue indicated by the pointer. So you know it's epithelial tissue because you see the free space and the cells are close together. Then you say, okay, multiple cell layers thick, stratified. Most superficial cells are flattened or elongated, squamous. The most superficial cells here don't have nuclei. They are essentially just sacs of keratin. Keratin is a structural protein and it's a waterproofing protein. It prevents water loss. So do the lipid granules in there, but that's another discussion. Keratinized. So if there's no nucleus in the most superficial cell layers, that is by definition keratinized stratified squamous epithelium. What does it make up? The epidermis, right? The entire outer portion of your skin. When you touch somebody, you are touching keratinized stratified squamous epithelium. Now, stratified cuboidal epithelium, you find it lining the ducts of glands, specifically sweat glands. So if you were to see this stratified because it's multiple cell layers thick, but notice that the deeper cell layer is kind of compressed or flattened. You don't ever look at the deep cell layer, you look at the superficial cell layer that's closest to the free space, in this case the duct, And this is the duct of a sweat gland. So down here we have the sweat gland. The sweat gland itself, the secretory portion of the gland, is going to be made from simple cuboidal epithelium. Then the duct that transmits that sweat from the gland to the surface of the skin, this duct here is lined by stratified cuboidal epithelium. Stratified because it's multiple cell layers thick. Cuboidal because the superficial cells are shaped like cubes. And epithelium for all the reasons it's called epithelium. It makes up the ducts of sweat glands. So what would be flowing through this little region right here would be sweat on its way to the surface of the skin. Only 2 layers, so it's going to be pretty easy to distinguish from stratified squamous epithelium, which always has multiple layers. Transitional epithelium. Transitional epithelium is a special type of stratified epithelium. You find it anywhere you need a lot of stretching to be able to occur. So you find it lining the ureters, which are the muscular tubes that transmit urine from the kidneys to the urinary bladder, and you find it in the urinary bladder. Why does the urinary bladder have transitional epithelium in it? It's because transitional epithelium can stretch and then return back to its original shape. But when it stretches, it takes on a slightly different appearance. So in a relaxed bladder, right, there's no stretch on the epithelium, and the most superficial cells almost look like globs. So notice there's no cilia up here, and they kind of look like blobs, right? Then when it's stretched, which I would never show you on an exam because it would be too tricky, it takes on an appearance, or it transitions from looking like one tissue to looking like another. Now it looks a lot like stratified squamous epithelium. If I give this to you, I'm going to give you the relaxed bladder, right? Meaning that those cells up top are going to look kind of like blobs, and they're not going to have cilia popping out of them. Where do you find this? Well, we have the ureters, which transmit urine from the kidneys to the urinary bladder, which is a muscular tube that stretches and then relaxes once you void that urine out of it. Thanks a lot and have a good day.